Hello students, welcome you all to a new video of Sunanda's tutorial. In this video we will discuss the stage 2 or step 2 of protein synthesis, that is translation in case of prokaryotes. So translation in prokaryotes is a cellular process by which ribosome, the organelle ribosome, synthesize functional proteins. So what is the step 1 of protein synthesis? So step 1 is transcription, where the DNA is converted into messenger RNA. Then messenger RNA by the process of translation is converted into polypeptide chain or protein. So because prokaryotic cells lack nucleus, translation is uniquely coupled with transcription. In case of eukaryotes, the transcription takes place inside the nucleus, and then that mRNA exits the nucleus, and then in cytoplasm the translation takes place. But here the prokaryotic organisms lack nucleus, so transcription is coupled with translation, meaning ribosome begin translating mRNA while it is still being synthesized by RNA polymerase. Okay, so DNA synthesizes mRNA at the same time. mRNA is also converted into the proteins, polypeptide chain. So both the process is taking place side by side. So what are the stages? At first, activation of amino acids. Actually, the DNA is first converted into mRNA. So genetic codes are there in mRNA. There are many amino acids which are linked with that particular genetic code. So at first those amino acids should be activated, okay, followed by tRNA activation or transfer RNA activation. Before translation begins, amino acids must be attached to their corresponding tRNA molecules. Okay. Enzyme which catalyze this process is amino acid tRNA synthetase. Okay. The energy this process requires ATP to link amino acid to 3' end of tRNA, forming activated or charged amino acid tRNA. So amino acid first linked with ATP, then it will form amino acid AMP by removing 2 phosphate groups. Then it will link with a specific tRNA at their 3' end to form charged transfer tRNA, or charged transfer RNA or tRNA. Okay. Next, the first step, which is initiation. The first step of translation is initiation. Yes, the translation process is divided into 3 phases or 3 stages. Initiation, next elongation, third one is termination, which is the final one. So initiation ensures that the ribosome is correctly aligned with the start codon of mRNA. Ribosome is the main machinery which helps in protein synthesis. So at first that mRNA is attached with the small ribosomal subunit. Okay. The 30S. So prokaryotes have 70S ribosome and the smaller unit is 30S. The 30S ribosomal subunit binds with purine rich region on the mRNA which is synthesized during transcription, and that particular attachment region is called Sine-Dalgarno sequence, which is purine rich region. Purine rich region means rich in adenine and guanine. This is guided by the initiation factor IF1 and IF3. This attachment process is guided by initiation factor 1 and 3. Okay. Initiator tRNA, the specialized initiator tRNA brings N-formyl methionine F-mate, the distinct first amino acids used in all bacterial translation. So the first codon is methionine, which is AUG. So the start codon is AUG, which codes for methionine. Then IF2 or initiation factor 2 bound to GTP or guanosine triphosphate escorts F-mate tRNA to start codon at ribosomal P site or peptidyl site. Then that tRNA along with methionine comes to the P site of the ribosome. Then GTP or guanosine triphosphate is hydrolyzed. The initiation factor dissociates and 50S ribosomal subunit locks into the place to form 70S initiation complex. So smaller ribosomal unit is there already. Now 50S ribosomal subunit locks or attaches on that place, forming 70S initiation complex with tRNA with that start codon methionine. Next, elongation, which is the stage 2 or step 2. Elongation is rapid and cyclical process where polypeptide chain is built one amino acid at a time. Okay, the ribosome utilizes three structural pockets: A site, amino acid site, peptidyl site, E site. Okay. A site, P site, and finally exit site. Okay, it is the larger subunit. Sorry, it is the smaller subunit. It is the larger subunit in case of prokaryotes, and it is the initiation complex, 70S initiation complex. Next is codon recognition. Elongation factor EFTU bound to GTP, guanosine triphosphate, delivers the next charged tRNA after tRNA F-mate or methionine, next charged tRNA to the vacant A site. After the shifting of tRNA along with methionine codon to the P site, now A site is vacant. So the next charged tRNA comes to the vacant A site. If the tRNA anticodon matches the mRNA codon, GTP is hydrolyzed and the elongation factor TU is recycled by elongation factor TS. Okay. Next, peptide bond formation takes place. The 23S rRNA or ribosomal RNA is a part of 50S or larger subunit of ribosome. It catalyzes a peptide bond between amino acid of P site and A site. That means one amino acid with the other. Peptide bond is formed between two amino acids. This transfers the growing peptide chain to tRNA in the A site. Okay. Then translocation. Elongation factor EFG utilizes GTP energy to push ribosome forward by exactly one codon along the mRNA direction or 5' to 3' direction. This moves now the empty tRNA to exit site and shifts the peptidyl tRNA from A site to P site, opening the A site for the next cycle. Okay, so this is how it is taking place by shifting one amino acid to the next, and peptide bond is formed between two amino acids. Along with that, the polypeptide chain is also formed slowly. Okay, next, the final stage, which is termination. Translation process finishes when ribosome encounters one of three stop codons. The start codon is AUG, okay, methionine, but the stop codons are UAA, UAG or UGA at A site. When the ribosome finds any one of these three, the translation process stops there. Then release factor. Because stop codons do not bind to tRNAs, specific proteins called release factor RF1 and RF2 binds directly to the A site. Then the protein release factor prompts the peptidyl transferase to break the chemical bond holding the polypeptide to the last tRNA, freeing the new completed protein or polypeptide chain. Then ribosome recycling, which is assisted by RF3 or release factor 3, and ribosome recycling factor RRAF. The mRNA and unchanged tRNAs detach and the 70S ribosome separates back into 30S and 50S subunits to be reused again. Okay. So this is the diagrammatic representation of the whole process, how this whole translation process takes place. So this is how the prokaryotic translation takes place. And with this I am concluding my lecture video here. I hope this video will be beneficial for you all who are in BSc Botany and MSc Botany. And please, if you liked it, please hit the like button, share with your other friends so that they can also get the benefits, and please subscribe to my channel Sunanda's Tutorial to get more valuable videos like this. Thank you.